Les voy a hacer una breve presentación de Mathilde, que es una ecóloga que está interesada en la ecología evolutiva, fisiología de la conservación y ecología de la conducta. Su interés principal es cómo la abundancia o deficiencia en micronutrientes afecta el desempeño en animales terrestres. Realizó su doctorado en el Instituto Multidisciplinar Hubert Curien, que es parte del CNRS, en Strasbourg, en Francia, donde estudió el impacto de la deficiencia del triptófano y la vitamina B3 causado por la generalización de monocultivos sobre el éxito reproductivo del hámster europeo, un vertebrado amenazado de extinción. En su postdoctorado está investigando los efectos del triptófano y la vitamina B3 en la ardilla listada, así como los efectos de deficiencias en estos micronutrientes en polinizadores y liebres, entre otros animales, además de intentar buscar maneras de contrarrestar los efectos negativos de los monocultivos. Pues como ven, tiene un interés muy diverso que se ajusta muy bien a los intereses de los investigadores aquí del instituto y estudiantes. Pues muchas gracias por aceptar. Thank you for accepting our invitation, Mathilde, and the floor is yours.
Thank you so much for the introduction, Alejandro, and thank you for having me. I'm really happy to be here with you today. So I'm going to talk about part of the work that I've done on tryptophan and vitamin B3, but I'm also going to talk about part of my work that I conducted in Quebec on chipmunks. So part of the work will be on conservation biology, and part of the work will be more on fundamental research and life history strategies. But before starting, I want to take a moment to acknowledge that I currently live in Quebec, in southern Quebec, Canada, and that my university is located on the unceded territory of the Abenaki people and the Wabanaki Confederacy, and that they are recognized as custodians of the lands and the waters of this area. So, as you all know, human population is growing, so we've been increasing the population from 600 million people to now almost 8 billion people, and this has major effects on our landscapes and our ecosystems. So five main global changes occur with climate change that we talk a lot now, but also pollution, invasive species, habitat loss, and overexploitation of natural resources. In terrestrial ecosystems, the main causes of global change are urbanization and conventional agriculture that are, both of them are changing the functionality, the diversity, and the management of the ecosystems. And by changing these ecosystems and their composition, their functionality, and their diversity, human activities are also altering the food that is available for wildlife. So we are impacting the availability of different food in different terrestrial ecosystems, but also the quality and the diversity of this food. Many wild animals are known to be constrained and restricted to monotonous diets in many monocultures. So here I present the one that are really common in Canada, but of course this is also observed in many other areas of the world where monocultures can be also including trees monocultures. And these animals are known to then have a diet that is lacking some essential nutrients. These monotonous diets are affecting many parameters related to animal performance, including their reproduction and their survival, but also their body condition and their behavior, ultimately their resistance and resilience to external threats such as pathogen exposure, climate change, or pesticide exposure in some cases. And in the end, it affects population dynamics with, of course, interrelations between all these different traits. So as Alejandro said, I'm particularly interested about the nutritional balance of animals in such conditions, but not only the intake that they have in carbohydrates, proteins, and fat that has received a lot of attention, but also the intake that they can have in some key vitamins and amino acids. I will do a focus on the vitamin B3 and its precursor, the tryptophan, for many reasons, and I will explain why during this introduction and during my presentation. So just so to introduce a bit tryptophan and vitamin B3, they are both essential in all animals, from invertebrates to all vertebrates, including humans, of course. And we must obtain them from the diet on a daily basis. They are very important for many behavioral traits, but also for modulating appetite and metabolism, because they are essential for the synthesis of key neurohormones and neurotransmitters like melatonin and serotonin that are affecting a lot of different behaviors. They are also at the basis of the synthesis of ATP and thus modulating the metabolisms. And deficiencies in vitamin B3 or tryptophan have been investigated a lot in humans, in domesticated animals, and in livestock. In humans, the deficiency in tryptophan and vitamin B3 is known to cause pellagra, a disease causing dermatitis, which is skin problems, diarrhea, and dementia. And in dogs, it's creating the black tongue syndrome. And in fish, sorry, I forgot to launch my timer. Oh, it's okay. And in fish, in salmon, domestic salmon, it's known to cause thermic stress because of an imbalance of the metabolism. Then in several domestic species as well, we can see problems of growth retards, as in calves and in rats, and aggressiveness in some rodents. And finally, in the domesticated honeybee, it's been shown that the deficiency in tryptophan and its derivatives causes problems of orientation in space. But what about wildlife? Well, we have very few information about wildlife for many reasons, especially that it's really hard to measure tryptophan and vitamin B3 in the food and in the tissues. So most of the studies have been conducted on domestic animals for that. But we know that wild animals are facing many threats, especially in farmlands, including pesticide exposure, problems of mechanization, but also this homogenization of the landscape, as I told before, with the generalization of crop monocultures. And this homogenization of the landscape has many effects on the ecology and the performance of animals. It leads to the lack of shelters to protect from predators and from the extreme of the weather. So animals are more and more confronted to these extreme weather events that are also increasing with climate change. They lose also their reproductive sites for many of them. And finally, there are a problem of foraging and starvation. And all these issues have been investigated a lot in birds and in pollinators, but of course, many other species are affected as well. And I will focus part of my work, part of the presentation, sorry, on the common hamster, which is not common anymore, unfortunately, but it's a species that is found in Europe and part of Asia and Russia as well. And so the common hamster, as I said, is not common anymore. It's critically endangered. It went from list concern to critically endangered in 2020 because of the decrease that has been observed in the past decades. So here you have a map where you can find the species. You can see in orange or yellow the relictual population that you have. And the species has disappeared from some countries now, like the Netherlands. And in France, it's hard to see, but you have a relictual population here in the northern part of France. And it's where I conducted my PhD under the supervision of Yvan Drick and Carolina Boll at the CNRS. And I will present you some of the studies that we've conducted on the effects of those monocultures on hamsters. So just to show you a bit what the landscape looks like in this area. So it's dominated by corn and wheat. And here you have two of my colleagues that are in a field where they are preparing to catch hamsters, so to place traps. And you have a drone that is going up. And you can see, so this is a cornfield. And then you have the edges and the roads, and you have in green the wheat fields and the city. So you can see like this conflict of land use between urbanization and agriculture. And as you can see, there is not a lot of edges or wildflowers. It's mostly bare land where corn will be sown and wheat that is growing, winter wheat. And so when hamsters emerge from hibernation, because they are hibernating in winter on these fields, they emerge mostly on bare soil. And the size of one field is 7 times the home range of a female. So they have mostly nothing to eat. And because they are hibernating, they are storing a lot of food in their burrows. They are not hibernators that will accumulate fat, so they can at least save the seeds, the grains of wheat and corn. And so what we started to do is to investigate the effects of corn and wheat on the reproduction and the survival of hamsters. So we fed hamsters with either wheat or corn, and we supplemented them with worms or clover because they are omnivorous, and so in the wild they will always complement their diet, their grain-based diet, with invertebrates or with other flowers or plants. And so here you have the composition of those diets. So if you can see from the left to the right, you have the wheatworm diet, with the same amount of proteins than the last one, which is the cornworm diet in red. And so you can see that these two diets, wheatworm and cornworm, are quite rich in proteins, whereas the two diets supplemented with clover were more rich in carbohydrates. So if you base your predictions on those observations, you will expect that the females fed the wheat worm and cornworm will have the same, similar reproductive success. It's actually not what we observed. They had the same rate of parturition, so they all gave birth to approximately the same number of pups. But then if you look at the survival of the pups, you have here on the top the wheatworm diet, the survival of the pups in the wheatworm diet. And then the three other diets, the survival rates of the pups was really low. And so what we looked at the beginning was the body condition of the mothers by looking at their body mass. So here you have the body mass on the second graph of the mothers during the experiment. So it's before giving birth, middle it's like halfway after giving birth, and end it's the weaning period. And as you can see in the red on the top, the female fed the cornworm, they were the one with the bigger or the higher body mass. And even though their pups, they all died. So we also measure other parameters. I won't show everything today, but we measure the maternal hormone, the oxytocin, which is linked to maternal attachment in mammals. And it was really high in this group as well. So we failed to explain why their pups did not survive, because their diets had a lot of proteins. They were in good body condition, even though their pups did not survive. And for the two diets supplemented with clover, we can explain that by perhaps a lack of protein in the diet. And then what I can also tell you is that these results were not explained by pesticide residues, because we measure them, and lack of minerals. So we've done for a year different measurements to try to explain those results, so including the pesticide residue, the different metals that we can find in the seeds, and the minerals. But actually they were higher, the metals and the pesticides, in the wheat, so it did not explain the results. At that time, I didn't know anything about vitamin B3 and tryptophan, but this is where I found the studies in humans. So our hypothesis was that maternal infanticides in hamsters and the low survival of the pups were caused by a deficiency in an essential amino acid, but we had to find which one. And so we found these studies. I found in the old literature in humans the impact of the consumption of corn, and these sentences, the musty corn and the dread-scourged pellagra, are the cause of a mysterious epidemic, which was at the beginning of the last century. And as perhaps some of you know, pellagra has been a scourge in Europe and in the USA for 2 centuries. And it was a disease that affected mostly poor people, from whose the diet was composed mostly of corn and dry meat, and where the corn was not cooked properly to free the vitamin B3. This disease, pellagra, is called the 3Ds disease, with dermatitis, diarrhea, and dementia. And if it's not cured in time, then it will lead to the death of people. And it's mostly caused by a deficiency in vitamin B3 in corn and its precursor, the tryptophan. So then when we saw this information, we conducted another study, because also it was linked to what we observed in hamsters, because on top of the low survival rates of the pups, we also had females that showed behavioral disorders. So they were not displaying any maternal behavior. They were storing their pups with the food, which indicated a cognitive issue. They were not taking care of the pups. They were not giving birth in the nest. they were not considering their pups as their progeny, but as food. We also observed some diarrhea, and with these maternal infanticides, on the only pups that survived, we observed skin problems. So based on that, we conducted another study. Here you have the results of the first study. So it's the number of pups that were weaned per group. And so in the other study, what we've done is just to supplement the cornworm group with vitamin B3. We did not want to redo the entire experiment because it's not really ethical to do such kind of experiments once you know what is the outcome. So we reduced as much as possible the groups, and we supplemented the cornworm group with vitamin B3, which restored completely maternal behavior and reproductive success, as you can see on this graph. So this is the cornworm group that we reconducted the second year with the cornworm and just the vitamin B3 that was added daily in the diet of hamsters. So just this addition of vitamin B3 completely restored the reproductive success of hamsters. But then the question that remained was to know whether hamsters were able to compensate for such deficiency in the wild by eating other plants or invertebrates, although, as I showed you, there are not much availability. So we conducted another study, this time in semi-wild conditions. So it was an experiment in enclosures, as you can see on the picture on the left. And this study lasted 5 months, and we had different groups. So we had hamsters that were released in wheat-only enclosures where we had sown wheat. Some of them were released in corn-only enclosures, and the other one were released in an enclosure with a mix of wheat, sunflower, corn, and alfalfa. And so what we've done also is that you can see a net on the picture, a black net on the top to prevent from predators. So everything was controlled and closed. There were no chemicals. We used organic seeds, and there were no predators. So it was only the diet that will explain the results that we observed. And we supplemented hamsters with the seed that was related to their group. So in the wheat enclosure, they received wheat seeds and corn seeds in the corn enclosure to be sure that they had enough food and that they were not limited by the size of the enclosure. And then we monitored the hamsters for 5 months with camera traps, with cameras as well, with capture, mark, recapture. And we recorded the species richness of adventive plants and the abundance of invertebrates. And finally, we monitored hamsters' reproductive success and survival. And so what we observed in terms of plants and invertebrates, so you have the 3 groups, the corn only, the mix, and the wheat. And for both the wheats or adventive plants and the invertebrates, we observed that the mixed enclosure were richer or had a higher abundance of invertebrates. And it's only in a 5 months period. So you can see that only sowing corn and wheat in such a time frame can limit the presence of other plants and invertebrates. And regarding the reproductive success of hamsters, so you have the number of pups per couple in the enclosures, and you can see that it was much higher in the mixed enclosure. So we had an average of 3 to 4 pups per couple, and in the corn only and wheat only, we had a very low reproductive success. So this suggests that they were not able to compensate the deficiency in corn and wheat by, well, in corn mostly, but it seems that there is also a limitation with wheat by eating other items in these enclosures. And this is not considering predation, as I said, or pesticide exposure. Also, this low reproductive success was associated to a high mortality of the adults after the first reproductive event. They died quite quickly in those monocultural enclosures. And so this highlights that one-crop enclosures and monocultures, again, are not suitable and of low quality for wildlife, even though when it's organic and even though you don't have predators. And this question also a conservation measure for the hamster, which consisted of having unharvested wheat, where we released every year hamsters in these enclosures. So these fields were fenced, and we collaborated with farmers that did not harvest the wheat. And so as a conservation measure for the species, we released individuals to sustain wild populations in these unharvested wheat. But then if this is not adapted for the reproduction nor the survival, then we have to change this kind of conservation measures. So the next step was to find crops with agroeconomic interest and high content in vitamin B3 that could compensate the deficiencies in corn, but that were also interesting for farmers to grow. So we started a project to look at the content in vitamin B3 and tryptophan, but also we realized consultations with many farmers in the region to have recommendations of the crops that could be interesting for them. And many of them suggested soybean, but others said that they used to grow sunflower in the area, and knowing that sunflower was quite rich in vitamin B3, we tested the association between corn and sunflower, corn and soy, wheat and sunflower, and wheat and soy, this time on a longer period on hamsters during their hibernation and their reproduction. I won't show all the results, just the conclusion because of a lack of time. But what we found is that wheat, soybean, and corn sunflower were adapted for hamsters' hibernation and reproduction, whereas corn, soybean, and wheat sunflower were not. So that's really interesting because there is these results show that there is not one favorable crop for sure. Soybean is good when associated with wheat, but not when associated with corn. We had a lot of behavioral disorders and infanticides in the corn-soybean group. And sunflower is positive when associated to corn, but not to wheat. So once again, it's more the complementarity of the crops that will be of interest for us than only having one single food. This project has been conducted as part of a very big project financed by Europe with many partners, including farmers, including different NGOs, policymakers, to try to preserve the species. But then the question that arises is how to expand the scope of this work and to transpose it to other systems, because as I told you, hamsters are now disappearing, and they are just a relictual population across Europe and part of Asia and Russia. And so it's perhaps not the best species to generalize such work, but also they are eating the crops. So even though we have some farmers that are inclined to protect them, most of them will not be interested in hamster conservation per se. So this is when I started to work on bumblebees with some colleagues from the south part of France. And we were interested in understanding how tryptophan and vitamin B3 deficiencies could affect bumblebees, because there are no studies looking at it, and also because they are a social insect, so we were not expecting to have the same impact of those deficiencies than what we observed in hamsters that are territorial vertebrates. Also, even though corn is wind pollinated, it can compose up to 50% of the diet of bees, including honeybees, but also some wild bees in summer in Europe. And it's been shown to reduce the lifespan of the bees. And finally, another point that I think is important is that there is another plant that is as deficient in tryptophan and vitamin B3 as corn, and it's the dandelion, which is also very present in the landscapes, mostly in the spring. So if you have a spring with a lot of dandelion and then a summer with a lot of corn in the landscape, this can be really detrimental because of the deficiencies, or at least this is what we wanted to try to look at. So we also conducted an experimental reduction of the dietary tryptophan and vitamin B3 supplementation in captive bumblebees. And just to show you briefly, what we show is that the reduction of tryptophan in the diet, so it was experimental in a sucrose diet. We control all the amino acids and the vitamins inside. And here you have three groups of reduced tryptophan, so low, medium, and high reduction of tryptophan. And the B3 supplementation is the same than the high reduction in tryptophan, so the last line of the graph, but we just added vitamin B3. And so if you look at this, this is the survival probability during the experiments, and just the addition, well, tryptophan reduction deficiencies reduced the probability to survive, but just the addition of vitamin B3 compensated that. And we also looked at the aggressiveness and the behavior of the bumblebees, and contrary to what we expected, we observed high rates of aggressiveness in venning bumblebees. So here you have just a short example of bumblebees fighting. It's really an extreme attack, it lasted more than 20 minutes and the attacker died at the end. So it was not a normal behavior to observe in bumblebees in such microcolonies in the lab. But also we show that adding vitamin B3 reduced the aggressiveness. So we've conducted several behavioral tests and we managed to see that vitamin B3 compensated that. And what is interesting is that the vitamin B3 level that we provided as a supplementation was mimicking the vitamin content, B3 content of sunflower. So this is where we started to investigate the potential benefits of sunflower, not only for hamsters as a compensation of corn, but also broadly for bumblebees. And this is where I started to work in Quebec on bumblebees. With Valérie Fournier from Université Laval and Lynn Adler from University of Massachusetts on the benefits of sunflower for bees. Mostly because also Lynn Adler, she's a researcher in Massachusetts working on the interaction between plants and pollinators. And her team has revealed that sunflower pollen has an important medicinal values for bumblebees. With many different experiments, they show that bumblebees that are fed sunflower pollen can get rid of this parasite, which is called Critidia. It's an intestinal parasite, a trypanosome, that is reducing the survival and affecting the behavior of the workers. And they show that when they fed bumblebees that were infected with these parasites with different pollen, they show that the sunflower fed individuals ultimately reduced completely their parasite load, sometimes to zero, which shows the importance of this pollen as a food resource for bees. And so Lynn Adler is working to inform beekeepers and farmers in Massachusetts. And what we've done after that is to implement a study in southern Quebec following these findings in the labs and in controlled environments. And we conducted a pilot study in Quebec in 2020 with 5 different sites. So we had 2 sites with only corn and no sunflower present in the landscape at more than 1 kilometer. Then we had 1 site with sunflower in the middle of the corn. And finally, we had 2 sites with only sunflower in the landscapes. And so what we found from this gradient of sunflower in the landscape, so it was a study, of course, in collaboration with many people and also with farmers. And so our goal was mostly to implement a non-invasive method to assess bee health in the wild, but also to assess the benefits of sunflower in the field and in the landscape. And so I don't know if you know about this, but the conventional method to assess bee health is still really invasive because it consists of capturing the bees from 30 individuals when you do genomics to 200 when you do other kind of analysis, and then you kill them and you extract the gut or you green them to obtain DNA or RNA depending on what you're looking at, and then you're conducting the analysis under the microscope or the PCR, conducting PCR molecular analysis or genomics to have any information on the viruses and other pathogens. But this method is not sustainable if you do conservation, and so we wanted to have a non-invasive method. And so what we've done is to validate this non-invasive method by implementing something that I've done in the lab with the team in Toulouse. So we capture the bees with net, we place them in a petri dish for several minutes in the shade, so they don't die because of the sun. And then we collect the feces and we can release the bees. And once the feces are collected, we can analyze the parasite loads under the microscope. And so using this method, we were able to do this fecal screening and to identify the parasite Critidia. So I cycled in red here. It's not really visible, I guess, but it's just to have a visual support of what I'm saying. And we also identified another pathogen, another parasite, which is Nosema. There are different species. We looked at Nosema bombi, which is found in bumblebees. And these are the spores of this pathogen, this parasite, sorry. And so finally, we could also confirm that sunflowers seem to have an effect, or at least that we found that the parasite load, so the presence of Critidia and Nosema, was more important in corn-dominated landscapes than when sunflower was present. So as you can see here, there is a greater variation, but also higher parasite load in bees. This is really preliminary because we didn't have a lot of field, and of course the landscape can have an effect, but it's confirming as well previous results. And so if I want to summarize a bit what I've just presented to you, if you look at the contents of the plants in tryptophan and vitamin B3, so we have the deficiencies that I told you about. So dandelion and corn are really deficient, and then most crops do not have, or at least most cereals and protein-rich seeds do not have a high content of vitamin B3. We have interesting contents in squash or pumpkins and sunflower. But then if you go to trees, you have really some trees that have really high content in those nutrients. And this is what I will focus on a bit later. But if you look at the deficiencies then in corn, we've shown that it induced behavioral and reproductive disorders in 2 species. We also have some results on brown hair, in European brown hair, but I won't present them today because of a lack of time. Then if you go to sunflower, we show that it increased maternal behavior and reproductive success when associated to corn in hamsters, that supplementation in vitamin B3 linked to sunflower reduces aggressiveness in bumblebees, and then that the pollen of this plant improved the health, or at least the disease resistance of some bumblebee species as well. But then if you go to the extreme end of the axis, you have the red maple. And this plant is the richest we found in terms of its content in vitamin B3 and tryptophan in all the plants that have been analyzed. And this is where it gets me to the project that I've done with different profs in Canada, in Quebec, so Patrick Bergeron, Dany Garand, and Denis Réal, who are monitoring a wild population of chipmunks in the southern part of Quebec, so in the red square you can see here. And in this population, not hamsters, switch to chipmunks. Chipmunks live in forest ecosystems that are dominated by the American beech and the red maple. And so there is 15 years of monitoring on 4 different sites. It's the same population. And it's a really complex system because chipmunks do not reproduce every spring, every year at the same time. Their reproduction actually depends on the mast cycle of American beech trees. So these are the seeds of American beech that chipmunks store in their burrow and that they rely on to reproduce. And when we talk about masts, it's because the American beech do not produce seeds massively every year. Here you have the graph shows the production of the seed production of beech every fall. And as you can see, for instance, in 2007, there were no production at all. The same in 2012. Sometimes there is a small production, but we call a mast when there is a high production, above 50 seeds for this species per square meters. And it's really when the resource will be so abundant that all the ground will be covered by seeds. And interestingly, chipmunk synchronize their breeding with the mast of American beech. So here, the histogram, the black histograms, are the reproductive success of chipmunks. As you can see, there is all the time there is a histogram just before the mast and just after the mast. So there is a reproduction in the summer before the beech mast that occur in the fall, and in the spring after the beech mast. So after hibernation, chipmunks reproduce again when there was a mast in the fall. But if there is no mast, there is no reproduction, neither before or after. So summer and spring. So this has this raises an interesting question: is that the chipmunks that reproduce in the summer before the mast, they will initiate reproduction 3 months before the mast occurs. And the chipmunks that will reproduce in the spring will reproduce 4 months after mast. So this raises the question that how can chipmunks anticipate, if they do, how can they reproduce 3 months in advance and before the mast? and mostly how can they synchronize juvenile emergence with seed abundance, because the female reproduce and the juveniles will emerge when the peak of seed abundance will be available. And then what are the ecological and evolutionary consequences of being born in spring or summer for chipmunks? So something interesting is now the link with red maple, because red maple produces seeds in the spring, and most of the time the production of red maple occurs in the same year than beech, but they do not, their seed abundance is really not related. And as you can see here, this is the diet of chipmunks in the spring. And you have the different years, and every time you have a star, it's a year of beech mast. And you can see that those years, chipmunks, so it's in red, they eat mostly red maple in the spring. So you have the diet of chipmunks that is really dominated by red maple. So when you observe that and you see, okay, in 2006, 8, 9, etc., you have a lot of red maple that is ingested, and chipmunks are reproducing, and then they reproduce, and the mast of beech will arrive in the fall. It's just a correlation, and there is no anticipation. They just respond to the availability of red maple. But what is interesting in this is that it's not really a response to the presence of red maple, because the consumption of red maple is not related to the production of red maple. As you can see on this graph, red maple seed production every year, and here you have the consumption of red maple, and we found no link between the two. However, we found that the consumption of red maple was related to the upcoming beech seed production in the fall, which means that chipmunks actively consume red maple seeds in the spring if a mast of beech was going to happen. So what we don't know is what is leading to this active consumption of red maple. Where are they still already reproducing and they focus on red maple as a good resource, or is red maple consumption triggering reproduction? This is an open question and we don't have the response yet. But what we know on top of that is that the consumption of red maple is the parameter that will optimize reproduction. So these are 3 different graphs where you have the proportion of females that were in oestrus, and we have the same for their reproductive success, so the number of juveniles that they weaned. And we looked at what had the main effect in our models. Was there red maple consumption, production, or the beech seed production that occur in the fall? And the more important parameters of reproductive success in chipmunks was red maple consumption. So it seems again that red maple has a huge impact in chipmunks. And then I leave you with the open questions on this, but I want to highlight this interesting intercorrelation between 3 species. So first, red maple, beech, American beech, and the chipmunk. And 3 of them are important for the interaction to occur. So if there is only production of seeds by red maple, chipmunks will not reproduce. If there is only beech, chipmunk will not reproduce. And if there is not any, of course, they will not reproduce. And what is interesting as well is that this strong correlation between the mast of these trees and chipmunk reproduction put a high pressure on the on the life history strategies of chipmunks. So for instance, here you have the summer cohort, so the chipmunks that are born in the summer before beech seed production. And when they are born in summer, they have a first opportunity to reproduce the next spring, just after the hibernation. However, for the chipmunks that are born in the spring, so after a mast, they will have to wait 15 months to have a first opportunity to reproduce, and it will be a summer reproduction. And if you look at these 2 seasons of birth that are caused by the production of red maple and beech nuts, then you have interesting impact on the life histories of chipmunks. You have the summer cohort that has what we call a fast life history strategy or pace of life with an early reproduction, as I showed you just before, at 7 months of age. They also have a higher fecundity. It's a paper conducted by Pierre-Olivier Montiglio with Denis Réal. And they have fast exploratory behavior. So I don't know if you know a lot about this, or if everyone knows about exploratory behavior, but they are monitored in open fields. So we conduct this open field in wild chipmunks every year. We capture wild individuals, and we do some open field to have information on their exploratory behavior in a new environment. And if you look at this graph, so it's extracted from this paper, the age at first breeding opportunity, so depending on whether chipmunks were born in the spring or the summer, has an impact on their exploration from slow to fast. And you can see that the chipmunks that are born in the summer have a fast exploration profile compared to the ones that are born in the spring in gray that have a slow exploration profile. And these individuals born in spring, they also have a delayed first reproduction at 15 months and low fecundity. And recently, we also show some correlation between the season of birth and the telomeres. So telomeres are the extreme ends of the chromosomes that are protecting the coding part of the DNA from degradation. And they have been involved in the senescence or aging and in the risk of developing cancer. So I won't spend too much time on this because I don't have time, but I just want to show you that these seasons of birth that are determined by the production of these trees have a huge impact on the life histories of chipmunks. And here you can see the telomere length of the females and the males at their first reproduction. And the females that are born in summer, they have longer telomeres than the ones that are born in spring. So this is a result that we recently published in a paper. I won't spend more time on this. It's really complex to present and long. But what I want to highlight from this is the importance, again, of food availability and food diversity and composition on the life histories of animals. And so if we go back to this graph that I showed before. So you have here the plant's content in tryptophan and vitamin B3 from deficiencies to abundance. And the deficiencies, as I showed before, are really present in farming systems. And then you have some impact of higher abundance of these nutrients on the performance and the behavior of species. And at the extreme end of this gradient, we have the red maple that improves reproductive success, triggers reproduction, and seems to have an impact on life histories of chipmunks. And now the last part of the presentation is how can we apply this knowledge to wildlife conservation in Canada, because this is something that motivates me beyond everything, is the conservation of wild declining animals. So we developed this project with a lot of people. I show some of them here, but it's now a project that involves more than 50 persons. And it's a project on sustainable agriculture to find solutions for farmers and for bees. And this project is really built around this idea of nutrients availability and all the knowledge that we can have on vitamin B3, tryptophan, but all the other nutrients, and try to connect all the dots. So between the preparatory actions, so trying to find solutions to manage and to have concrete actions to preserve species in farmlands, and then concrete conservation actions to monitor the impacts that we have, the real impact that we have on species. And finally, some knowledge transfer dissemination to farmers and to the public to raise public awareness. So what we try to do first, for instance, in these preparatory actions, is to survey farmers really broadly, so to have more than several hundred people answering this survey. So what do they know about native pollinators? Would they be interested in integrating favorable measures in their farm? If so, what will be their main limits, time, space, money, etc.? And what would they prefer? So we will present to them different associations, different plants that we identified as favorable, and they will have to choose. The idea is to provide a diversity of options to farmers so they can implement the ones that are more adapted to their conditions. Then we also work on the identification of plants and plant associations of interest based on their nutritional and medicinal values for bees. So I just want to show you quickly this table. So it's a work from Roxane Cloutier, who is a student working with me. So I don't want you to look at the detail of the table. This would be really unpleasant. But you have this column with tryptophan. And you have here the content of corn in tryptophan, which is 0 or not detected, which means that the pollen of corn is deficient as well as the plant. And then you have other plants like the squash, but also the sunflower that have higher content in their pollen in tryptophan and vitamin B3. And so what we try to do now is to find the association. So based on all the nutrients, so we have all the amino acids, vitamins, etc., we try to combine the one that could be complementary. And because we only have the information in the literature for crops, we also develop a technique with a colleague from the University of Sherbrooke to measure all the nutrients in the pollen of native plants, so including trees, bushes, and wildflowers, to have the information on what plant is rich in the nutrients that are lacking in the crops that are grown in monocultures. And finally, when we identified some favorable pollen or pollen associations, we are validating their benefits, their nutritional and medicinal values for bumblebees in collaboration with an NGO. So we work on wild-caught queens because they have a bumblebee conservation lab where they capture wild queens and they try to reproduce them in their facilities to make them hibernate. Once they have done reproduction, the new queens that will emerge are called gynes, and these gynes will hibernate, and the idea is to then release new queens or gynes in the wild the next spring to sustain wild populations. And to do that, we are collaborating with some beekeepers. So I'm approaching to the end of the talk, and I just want to present a really preliminary results on the benefits of red maple again, this time for the reproduction of these bumblebees. So what we've done with this NGO and with Sarah is to feed some wild-caught bumblebee queens with red maple and a mix of white flowers. So they had 75-25 of each. And the control group only had the white flowers. And as you can see, this is the reproductive success of the queens fed red maple. And so we had 10 queens in each group, which is not a high sample size, but we will reproduce the study again. And on these 10 queens, they produced more than 1,400 queens, and approximately half in the control group. And finally, the control group failed to produce new queens, so gynes, whereas in the red maple group, we had 21 queens, which is like they doubled the initial population, basically. And the same for males that are produced at the end of the season. We have almost 4 times more males when the queens were fed red maple than when they were fed wildflowers. I just want to specify that they were fed red maple for only a month in spring, like the duration of the availability of red maple in the wild, and then they were switched as well to a mix of wildflowers as they will do in the wild. So I think I won't have time for the last slide, so just a quick overview, but we are also for these concrete conservation actions working with farmers in greenhouses to improve the sustainability of their approaches, because all the crops that are grown in greenhouses, well, at least most of them, they use commercial bumblebees to pollinate the crops inside the greenhouses. But these bumblebees come with some disease, and they spread the disease to wild pollinators because the greenhouses are not sealed. And so they can escape from the greenhouses and they interact with wild bees and they transmit viruses and different pathogens. And considering the medicinal values of the sunflower, we are developing these studies with farmers where we place sunflower in their greenhouse. And it was not easy at first because most of them said that they would not have space. But actually it worked really well because we used dwarf sunflower and we managed to put it everywhere we could. And so we worked with different farmers on this project and it worked really well. It reduced the parasite load of the bumblebees. This study will be reconducted in 2023. And to finish this time, we also develop now this mesocosm. So it's we will be able to do experiments for bee conservation and for sustainable agriculture and to manipulate the crops that we put inside and the water treatments. So the idea will be to compare corn and associations between corn and plants that have complementary values. And the ultimate idea is to find solutions that are economically interesting for farmers, relevant in a commercial production, but also resilient to climate change and that have benefits for the bees. So ultimately, my take-home message to finish is that we have this variation in tryptophan and vitamin D3 from important deficiencies in crops such as corn, and then we have plants that have really high content. And if we can work in trying to find the best associations, we can do that in the fields with farmers, but we can also do that in the landscape by using road margins and trying to associate in the landscape the plant that can be complementary so that wildlife and bees can have a diversified food and reproduce properly. So thank you. I hope I was not too long. And I want to thank all the partners and all the founders of this work and all the field crew that collected the data on the chipmunk project. And of course, the amazing chipmunk hamsters and bumblebees. Thank you for your time and your attention.